they make up what we call the renal corpuscle. This is the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Supposed to be an L. And the DCT is the distal convoluted tubule. And I'm not going to write it out, but you can see proximal convoluted tubule and distal convoluted tubule. Together, these three things make up the renal tubule. We have connecting ducts, and then we're going to have this thing here, which is called a collecting duct. The collecting duct is going to absorb or collect the waste dripping out of all of these nephrons. There's another nephron here and here and here and one going this way. They go all the way around in 3 dimensions. And they're really not flat and pretty like this. The way they would look is this. I have a Bowmans capsule. I have a proximal convoluted tubule, a loop of Henle, and a distal convoluted tubule very close to each other. But if I started to pull them apart, they'd be more like this. And essentially what happens is an afferent arteriole comes in, forms a glomerulus, and then the peritubular capillary bed is literally wrapped around all of the tubule, hence the name peritubular. Okay, so they really look more like this, but we've spread it out so it's all nice and pretty. So here's a few of the other names of structures that you need to know. This blood vessel coming into the glomerulus is the afferent arteriole. And if you remember from part 1 A and P, afferre means to carry towards. It's going to carry blood to the glomerulus, and out of the glomerulus, we're going to have the efferent arteriole. And efferre means to carry away from. It's going to carry blood away from the glomerulus, and then this capillary bed would literally be sitting over the tubule. So this is called the peritubular capillary, or capillaries. So as blood comes in through the arcuate artery, it'll go up through the cortical radiate artery, through an afferent arteriole, through the glomerulus, where we pull everything out, good and bad. Then or not everything, but some of the stuff, the efferent arteriole, and then it goes into the peritubular capillary. This tubule is going to select the good stuff, nutrients, ions, and water, and pull it back into our bloodstream, and just a little bit of water and waste comes out. We cover this in great detail in lecture. So this is the anatomy of one part of our model that we're going to go over. So I hope you have all of those structures down. We have the afferent arteriole, the glomerulus, the efferent arteriole, the peritubular capillaries, and then we're going to have our Bowmans capsule, proximal convoluted tubule, the descending and ascending limbs of the loop of Henle, the distal convoluted tubule, the collecting duct. Okay. Now, I'm going to zoom in now. What I'm going to do for our last drawing, I know this video is getting kind of long, but I wanted to cover all of these at once, is I'm going to cut out a section of all of this. And in reality, I'm going to draw something here. Our Bowmans capsule, our proximal convoluted tubule, our loop of Henle, and our distal convoluted tubule, where the capillaries come in and out from the glomerulus. Always where the efferent arteriole is, or between these two blood vessels, a piece of the distal convoluted tubule loops back right here before it takes off. So actually, this distal convoluted tubule would end up in part of this picture. In reality, that's how it looks. We don't draw it that way, and our model doesn't show it that way initially because it's too hard to follow if we did it in reality. So now I'm going to cut all of this out with a little piece of the distal convoluted tubule. I'm going to make our last drawing. And this is one of the hardest things for students to understand on this set of models. So, here's my Bowmans capsule. I'm going to use some of the same abbreviations that I used in the last drawing. So this is all Bowmans capsule. This tube coming out here is going to be our proximal convoluted tubule. I'm going to have a blood vessel here, and when you look at the model, this blood vessel has a lot of thickness to the wall. It's going to come in. This is our afferent arteriole. It's going to form a big knot of capillaries that are all interconnected. And then it's going to come back out and it's going to branch pretty quickly. And on this model, they cut a section out here where there's kind of an opening, and there's these tan-looking cells on here. And finally on the model, you're going to see a piece of the distal convoluted tubule. Let me make sure all of this is visible on my screen. Yeah, I'm drawing it a little bit high, so let me move my camera again. So, I have Bowmans capsule, proximal convoluted tubule, the afferent arteriole. This is going to be the efferent arteriole. That's our distal convoluted tubule up there. Now, the space in here, inside the Bowmans capsule, is called the capsular space. And when we filter stuff out of the blood, it will enter the capsular space, which will then drain through the proximal convoluted tubule. This is our glomerulus. On half of the model, if you look at it, there's all these little pock marks all over the glomerulus. By the way, those little openings, they're not on our list, but those are the fenestrations. If you studied blood vessels very well, you know fenestra means window, fenestrations are openings. We have fenestrated capillaries in our kidneys. They're only found in a few places: brain, kidneys, liver. So that's where the larger stuff is going to come out and be filtered out. Now, if you look at the other part of this glomerulus, you're going to see that it's covered by these little cells. And these cells have these interlocking feet. Now, they're kind of a tan color. I'm going to use orange, but you're going to see these cells that are covering the capillary. And they have these little, I don't know, looks like little fingers sticking out. And if I had another cell here, its fingers would interlock with this cell. And it's very hard for students to see this. But essentially, I have these funky looking cells that look like this. They look like little starfish. And they have these little finger-like extensions on their edges, okay? Those little finger-like extensions are called a pedicle. And these are called foot pods. And then there would be a nucleus, usually it's light blue. Now, this cell can wrap around a capillary. And if I take a second one, I'm going to use a slightly different color. But if I had another one of these cells, and one of its, here's the nucleus, and here's the branches. They can interlock their little fingers around a capillary. And when I interlock those fingers, if you see the blue of my shirt, let me do this so you can see it. If this is the nucleus, the blue spot is the nucleus of a cell, and this is another cell. When they interlock their pedicles, you can still see some of the blue of my shirt between my fingers. So that is a slit. If I tried to have something be shot out of me like this, if my shirt was leaking fluids through it, if there was large structures, they would be trapped by the pedicles and stay inside, and only the water would seep out. So these cells are called podocytes. The podocytes wrap around the capillary, and they create these little filtration slits. That's how we filter our blood. It's called the filtration membrane. So when I ask you to identify the cell, it's a podocyte. If I ask you to identify the little finger-like structures, they're called a pedicle. And then the gap between them, you're actually going to see the red of the capillary in between them. And all of these little red squiggly lines is where we would have what's called a filtration slit. So two sets of pedicles interlock. The gap between my fingers, if my fingers are pedicles, the gap would be a filtration slit. And then you would see my shirt through there. And that's really how we're seeing the filtration slit. Now finally on this model, this little cutout of cells is called the juxtaglomerular apparatus. I'm going to put JA as I point to that. It's this word. Juxta means next to. Glomerular because it's next to the glomerulus. And those are called the juxtaglomerular cells. Okay, so let me put JC. On the distal convoluted tubule, I have little cells going all the way around, a single layer of cuboidal cells. But if you look on one side, the side that's closer to the juxtaglomerular cells, it's a little bit thicker. That thick layer of cells are called the macula densa. If I put both of these together, they're called the juxtaglomerular apparatus. And what it turns out is the juxtaglomerular apparatus is it has pressure receptors, baroreceptors that we talked about at the beginning of the semester. And as blood is flowing in, there should be a certain amount of pressure squirting the fluid out here, so we're filtering our blood. As my blood pressure goes up and down, I cannot alter the rate at which I'm removing toxins. So if my blood pressure drops, these cells can signal to change my pressures so that I am filtering at the same rate. Also, as fluid is going through the distal convoluted tubule, we should have a certain amount of pressure. And these cells are detecting the pressures, and if they're not right, they can release the hormone aldosterone. If you don't remember what aldosterone does, aldosterone raises your blood pressure by increasing the reabsorption of salt and water. They can also release another compound called renin, and we'll talk about that in lecture. So this is the drawing of the model. It's the hardest thing for students to see is these podocytes, pedicles, it's called a pedicle or a pedicel. Some books use the term pedicle. I think ours use pedicels. Okay. So anyway, that is the hardest thing for students to see on this anatomy. So I like to draw these models out first. The next series of videos that I'm going to put on my Canvas page are going to be the models in correct order. So I know this was lengthy. I probably should have broken it up, but that's okay. I hope you had as much fun as I did. I'll see you in the next video.